you all for coming. It's a great pleasure today to welcome Professor Eric Boczko of Vanderbilt University, who has been a former colleague of mine, and he's one of the very unique individuals in terms of his training, in terms of his approach to biological and medical discovery, and in terms of combining mathematical, computational, biological, and medical techniques and reasoning in his research. I think he's one of the very few people in the country today that has this range and depth of techniques that he puts to practice in order to conduct research, as you will find out in his talk today. So Professor Boczko, in terms of background, earned his PhD in biophysics from Carnegie Mellon University, and after that he earned his second PhD in mathematics from Georgia Tech. Subsequently, he taught mathematics, and he ended up as a member of the biomedical informatics faculty at Vanderbilt University, where he is now associate professor, as I mentioned previously. His interests span mathematical biology, dynamical systems, bacterial physiology, and probabilistic causality. And today he will discuss with us his findings in his studies, where he studies bacterial dynamics and how they affect nosocomial infections. So, Eric, welcome, and we're very eager to listen to your talk.
Thank you. Well, thanks, Constantine, and it's a real pleasure to be here. I've known a lot of people, Alex and Constantine and Yin, Lawrence, for quite a while, and it's good to see everybody, and it's great to be able to be here. Like, what was so cool is that this is like a translational research place. We were at Todd and I we were at Cold Spring Harbor for a meeting, and some of the PIs, Todd sat at a table with some PIs that were grumbling about all of the NIH money that's going to translational research. And I'm thinking, yeah. So the work that I'm going to tell you about, the idea kind of behind this was that a few years ago, I wrote a grant proposal that I was in love with. I really loved it. I thought it was brilliant and fun and was sure to get funded, and it didn't. And I realized then it was about yeast, and that the NIH really wasn't going to fund stuff about, and someone told me this actually, I thought this is a great line, that the NIH is not interested in funding diseases in yeast. And I thought, okay, then you know, it's survival of the fittest, and I had better get fitter. And so I thought about ways, things that were interesting to me, interesting ways that I could get closer to the hospital and to people, but stay within the kind of areas that I was interested in, which was bacterial dynamics and physiology. And I thought to myself, this actually came because I would go to Health Plus, which is a exercise facility at Vanderbilt, and there was this other guy that would get there at 5 in the morning, and we were both competing for this one treadmill. That they had one really good treadmill that everybody wanted, and it was, you know, who would get there 5 minutes before the other guy. And I hate this guy. I really hated this guy, because some days he would beat me to this machine. And then the guy stopped coming, and I was like, yeah. And it turns out, and then there was a promotional video for this place, and it turns out that this guy's wife had some kind of debilitating disease, and he was taking care of her. He went into the hospital for a routine, like hernia kind of outpatient thing. He got an infection and died. And then, of course, you know, because this is a guy, like, I mean, I was like actively competing with the guy and was wishing for his death. You know, like that I felt terribly guilty about this whole incident. And I thought, all right, nosocomial infections are a bad thing, and that's a good place where I can start, maybe. And obviously, true story. It kind of, you know, we're all a little bad, right? So, okay, so then the real idea here was that the kind of thing, maybe the more reasoned thing here is that I started to think about this, is that you and I are imbued, we're very fortunate that we're imbued with an immune system that surveils our body all the time for foreign objects, right? And actually has the dynamic ability to sense pathogens and to kill them. And the deal is that buildings, spaces like the hospital, they don't, right? But it's well within, like, it's well within our cognitive purview that we can create intelligent spaces, right? Nowadays, you can't, no one, like if you go to a bar, if you own a bar, it's ridiculously foolish to paint the walls, right? What you have is small little cameras that project colored images onto the walls that you can change every 5 seconds, right? So you can, I mean, that's a very small example, and it's up to us to build more complex examples, but it's well within our consciousness to create intelligent spaces. So we can build, and control theory is a highly developed sort of subject. So it's possible that we can, if we understand a dynamical system, we can control it, and we can create intelligent spaces, and it's possible that we could, with understanding, imbue a hospital environment with an artificial immune system. But before we can do something like that, in my mind, I mean, what's my approach? You guys probably have lots of better ideas than I do, but in my mind, how do you approach something like that to undertake a task like that once you've conceived of it? To my mind, where it starts for me is that you need to understand the dynamics. You need to be able to understand the dynamical system that you want to control. If you want to keep planes from falling out of the sky, you need to understand you need a model for the vibrations of the wings, and then you can control these vibrations, right? I mean, that's what goes on. And in doing the kinds of things that I'm telling you about today, I'm always very surprised. I'm not a physician, and it's a good thing. But if I was a physician, I would behave very differently. When I look at how physicians do their job, I would do it very differently. And it's a real thing for me, because I would conduct medicine much more like a chemical engineer would control a process at an engineering plant, right? They work out the dynamics of everything, they optimize everything, and they have very tight process control over every step. And medicine isn't practiced that way. And I don't really understand. I mean, I'm sure that that's historical and it's not done maliciously or out of ignorance or something. I'm sure that there's a reason for that. It's just that when I look at it, I think, Jesus, that's just so bass-ackwards, you know? I wouldn't do it like that. So it's the things that I'm telling you about is really my own cognitive working through that disparity. And this is kind of how I would practice medicine if I was licensed to do so, which all of us are very fortunate that I'm not. So the first thing that we started out to do is to try to understand the bacterial dynamics and try to understand, you know, nosocomial infections are a big problem. And at Vanderbilt, and I think other places, you probably have run across this, that ventilator-associated pneumonia is sort of a central infectious hub. People get ventilated, people on the in the intensive care units, it's very likely to get ventilated. And once you're ventilated, it's very likely that you're going to develop a pneumonia. And when we started to look at organisms in the hospital, this is a graph that we made that tried to connect organisms and infections. And there's, you know, everybody's, and so in the course of starting to do some of these things, I found a direction. So this is how pneumonia is dealt with by physicians. These are the guidelines for how a patient presents when do you know that a patient has ventilator-associated pneumonia? These are the diagnostics. If there's some radiographic infiltrate, chest x-rays is a very blunt instrument for detecting infection. X-rays don't actually detect the presence of bacteria, they just detect water or infiltration of leukocytes. So sometimes the horse is out of the barn already. And then the deal is that temperature, what I liked about when I read this paper, what struck me was that the measurements that here are dynamic. You know, because I'm very interested in dynamical systems, and for me everything is a process. And if you're trying to decide if someone has pneumonia based on a point estimate, you know, like just one single number, this doesn't seem right to me. Really, it's a process, right? You want something you want to look at it, like, you know, somebody's hooked up to a heart monitor, you know when things are going south because you hear the beep, beep, beep starting to rise or crescendo or whatever, right? So the things here that are time-sensitive measurements, like you can take temperature continuously, or white blood cells, you can get those measures continuously. The problem with those measures is that they're nonspecific, right? Infections in your leg or your liver will trigger those kinds of changes just as easily as things in your lungs. So the thing that I read that kind of set me off on this direction was there is a thing in here that if people that had profiled for tracheal aspirates multiple times per week, if they took multiple samples and tried to estimate from repeated samples, they were able to predict pneumonias about 85% of the time. And that seemed to me like a good thing. And where I'm at Vanderbilt, the way that the process works is when a physician sees that there's clinical suspicion of pneumonia, they perform a quantitative BAL. Now, lots of people, you know, where I'm at, everybody argues about everything. So the physicians argue about the BALs, whether it's good or bad or whatever. But it is a quantitative technique where a person gets an invasive procedure, the tube gets put into the lungs, fluid is squirted in there, they pull fluid out, and then they culture that. And its sensitivity and specificity varies, which is what people argue about, so there's really no kind of gold standard for pneumonia. So along the way, kind of what developed for us was I'd read a couple of papers. Actually, this goes way back to something that I read in graduate school about the engineers that create microchips. In the rooms where they create microchips, the technicians are fully gowned, and they have you know, much more so than physicians are, because small respiratory droplets called Flugie's droplets will land on the microcircuitry and foul a chip and cost Intel or whoever lots of money. And the clean rooms in which, I don't know if you find this as preposterous as me, but this is one of those things that I find preposterous, right? So the clean rooms in which they manufacture microchips are so much more exacting in terms of cleanliness and in terms of like controlled environment than a hospital room. But you're talking about the difference between a microchip and a person, right? And it's a little absurd, right? I mean, we all know our value, don't we, by now? What's more important, a microchip or the megachip? I mean, it's the microchip, clearly. Anyway, so I'd read this paper where, you know, obviously these droplets are coming out and fouling chips. And then there was a recent paper in PLOS where they had found virus in exhaled aerosolized droplets. And also they used an electronic nose in a barn to find pneumococcus in horses. Horses also get pneumonia, and the horses actually infect each other by breathing on each other. And so there's virus in the air, and they could detect this with the electronic sniffer. So that gave me the idea that perhaps in aerosolized droplets that people are putting out there, we can detect bacteria that are in their lungs or viruses or whatnot. And this was kind of the scheme that we cooked up, which was people are intubated, they're breathing up these little droplets. If we can capture these droplets, we can do PCR and find the organisms, and we can do this repeatedly in time and look at basically growth curves inside of people. And today, that's the kind of thing that I'm going to tell you. Really, the greatest thing that kind of happened in this whole thing was that the respiratory therapist at Vanderbilt let me poke around the ventilator circuits, and we discovered that in the ventilator circuit, just outside the person's body, there's this small filter that contains a little sponge, and it's called a heat condenser humidifier. They're either called HCHs or HMEs, heat moisture exchange filter. The idea is that the bone dry air that's being put into the patient's lung would desiccate a person if you didn't remoisturize the airway. And some ventilators, they actually have a humidifier, but over the course of probably the last 10 or 20 years, people have, some places, this is not universally used everywhere, I've discovered, but a lot of places have gone to this filter, which very quickly gets filled up with aerosolized and condensed breath from the patient, and then heats and remoisturizes the airway. So this thing sits in line, and the respiratory therapists at Vanderbilt and any place else that they use them, every 8 hours they take this filter and they throw it in the trash. And that is brilliant from several points of view. Number one, you do not have to wrestle with the IRB to look at this, because once it's, see, when you study this thing, this is the technical stuff that a bioethicist told me once, and I like this, it's kind of cool, that once you study this, you're not studying the patient anymore, you're studying scraps. And so you can study this to your heart's content as long as you don't connect it to the patient. Once you do that, then you got to get, you know, the waiver of consent and all that. But just looking at this, it's scraps. And the thing that, the most important message that I can tell you today is that this thing is a gold mine. This is a gold mine reservoir of information. That's what I will show you, the data that we've done in the laboratory. And then there's some mathy stuff that we've done that, you know, is just a little icing on the cake, which I hope you'll like too, because actually I really like it. It's simple and it's interesting. But the real take-home message, the talk could end here, is that this is an incredibly rich reservoir of information about what's going on in people's lungs, ventilated patients. And there's quite a lot of them. Burn patients are ventilated, kids are ventilated, everybody's ventilated. So AirLife, who makes these things, won't tell me anything about them. I've learned quite a few things, though, on my own. So we figured out that they're coated with calcium chloride. That's what makes them, you know, there's that word with the G in it that I can never say, hydroscopic, so they hold on to water molecules. And it's calcium chloride in this non-latex sponge environment. When we do, so I will tell you that we have done some metabolomics, so GC mass spec or HPLC on the fluid that comes out of there. And what you find, so for instance, there's all kinds of uracanate, all kinds of plasticizers associated with the construction of the sponge. You can find those there, along with things like when we profiled some of these from patients, one of the things that popped up was levulinic acid, which is, I found out, an additive in cigarettes that makes the nicotine more potent. And that makes its way into people's lungs, smokers, and it is readily exhaled. So there's, okay, so our main goal was, well, now that we've found this thing, we need to optimize and understand how do we retrieve information from this. We want to retrieve cells and biomarkers. We want to do this quickly and accurately. And for right now, it's a research tool. This is, we're thinking about generating point-of-care devices based on LSPR sensors and so on and so forth. But that's a few years down the road, because obviously what you would like to have is a filter unit just like that that turns colors, right? That the physician can look at, you know, when it's loaded with bacteria or if it's loaded with cytokines, it turns blue. And if it's really dangerous environment, if there's a key biomarker, it turns red, right? And presumably, and what I'm going to talk to you about is how do we detect biomarkers of pulmonary infection early in the process. One of the first things that we wanted to look at is what kind of environment does this filter present to the bacteria. And what we did was we took We grew organisms on the bench. That's the thick black line. And we took an aliquot of that and put it in the filter, and then removed that aliquot back out of the filter every couple hours to see whether or not the bacteria were growing in the filter. So how is that environment bacteriostatic? And in fact, you can see from the graph that the thick black line is the bacteria on the benchtop keep growing and the ones in the filter don't grow. So the filters are bacteriostatic. And in fact, the filters are bactericidal as well. So if we look at the percent of replicating or viable cells, the ones on the benchtops stay constant, and the ones that we put in the filter stop replicating. So basically the filters are not only bacteriostatic, but they're bactericidal. So that made us happy. And then we did these experiments where we would put a certain preset amount of bacteria into the filter, and then we would try to see how much percent recovery we could have, right? And the most important thing here is not necessarily what your percent recovery is, it's what's the variability, right? Is if you're getting 10% and every single day you get 10% with small variability, bam. That, in my mind, that's perfect, right? Because then you're getting a representative, reproducible sample, and that's workable. And it turns out that the reproducibility, the variability is actually very low, and we are able to, after 8 hours, recover about 50% of what we put back in. Yep.
So in terms of the experimental setup with introducing the cells, did you try to replicate this sort of exhaling and inhaling, and just sort of model it, or was it literally you just stuck a needle in and injected?
Oh yeah. So it's like so these experiments that I'm telling you about are done in the least elegant, in the least physiologically realistic way. They're simply process experiments where we grew bacteria on a benchtop, we grew E. coli, we grew staph, we take a pipette man, we squirt 5 mLs in there, we parafilm them, put them in a refrigerator, take them back out, pull it out, look at it.
Solid sponge, right? It's like, there's not, it's just literally a tube, it's a sponge tube. And there's no like...
Yeah, this is exactly what it looks like. It's almost exactly the dimensions. I don't know how big it is up there, but over here it's about right.
It's uniform, it's shaped like exactly like a cylinder.
So this is like that thing. You remember in Armageddon, the dude strapped into the, what's the guy's name? Owen Wilson strapped in, and he's like, this is so exciting. We're like in this giant piece of machinery built by the lowest bidder, like he's about to get shot into space in a vehicle that's built by the lowest bidder. That's the deal, right? This thing is made out of chicken gristle and sold for like $10 a unit, right? It is the absolute most it's the cheapest sponge that you can make that's not FDA disapproved, right? Yes, so I think what you're asking me is that the sponge is homogeneous, and from lot to lot it's the same. So the experiments that you're talking about, I love chemical engineers, and Ken Debelak in chemical engineering and I collaborate a lot, and we've been trying to build a point-of-care device based on this kind of thing. And in doing that, one of the things that we've developed is an aerosol dispenser that behaves, that we can, that simulates a lung kind of environment. For this kind of stuff, I think that's overkill, but for down the line kind of things, I think it's important. So one of the great things about Vanderbilt is that they have a translational research institute headed by Dr. Gordon Bernard, who I'll thank more formally at the end. And I put in a small proposal to take this work, to start this work, and then to take it further. And they were very generous to give me some funding. And actually, I had submitted a very complex, convoluted design. And Gordon Bernard said, no, do it like this. And this is his design, which was very nice, which is very simple to kind of quantitatively figure out whether or not the things that we're seeing in these filters matches the kind of current gold standard, which is things that you'd find in BAL. And so Gordon's design was really simple. Every time in the intensive care unit that someone is slated to have a BAL, which means they have clinical suspicion of pneumonia, they usually do that on rounds in the morning. When they identify that person, they would replace whatever the filter was there with a brand new filter, and then it takes about 4 hours till rounds are over, till they actually do the procedure. So the filter was on for about 4 hours. They would then take the filter off, put it in a bag, perform the BAL, and then give me a sample of the BAL fluid, and so that I have time-matched samples from the same patient. And the filters were removed before the BAL so that the BAL process itself would not interfere with whatever data was there. And this was a brilliant design and not my own. And then what we did is we cooked up a way to look at just pathogen detection in these matched samples. We have primers against 7 of the most, I think the next, so there's lots of like accounting that happens in the medical record. So we looked at the medical record in these 192 SICU encounters in pneumonias. You can see this is kind of the distribution of organisms. And so what I did was I picked sort of the top 7 organisms that cover, you know, most everything that's going on here. And we also use a universal ribosomal DNA probe, so that acts like a checksum. So we have 7, we profile for 7 very specific organisms, and then for a universal organism. So if none of these guys, the specific ones, were to show up and our universal was off the charts, we'd know that we'd missed something, and then we can sequence the universal one and figure out who it is that we missed. So that was kind of the design of our thing, and these are the 7, 8 PCR targets that we had, 7 specific ones, some Enterococci, Staph aureus, which is the thing that we see the most often, Pseudomonas, E. coli, Acinetobacter, Klebsiella, and then we have our universal primers that pick up everything else. So here's, well, in 53 samples over 2 months, basically, we got 53 samples, and this is the gestalt of that. So the first column shows you the bacterial targets that we detected from the aerosolized fluid that collects in the HME. The second column tells you about the targets that we detected in the BAL. And those were done side by side with the same, you know, independently side by side with the PCR methods. And then the last one tells you about the culture results from the BAL that come back from the microbiology lab. And the microbiology lab does things completely differently, right? They don't use PCR, they do culture techniques. So the end result is that in 2 of these samples, there was some mismatches, and you can see those indicated by the stars. In one of them, there was about 100 to 200 cells per ml of Enterococcus faecalis, and that we missed in the HME. And again, in another filter, there was Staph aureus at about the same low level, 100 to 200 organisms per ml, that we missed in the HME. Other than that, yeah.
This is really cool. Anyways, I had 2 questions. The first one was, so I noticed that you selected sponges for patients that had suspected pneumonia to begin with. So if you don't have any suspicion of pneumonia, is there a baseline level of sort of, you know, when you breathe out, I just don't remember, is it sterile when you breathe out? I don't think it's sterile when you breathe out. So is there a baseline sort of, you know, level of bacteria? And then my second question is, with your thresholds that you set for, you know, detection, the PCR detection, was there sort of some cutoff point that you said that if it amplified X amount, then we'll consider it to be there?
So there, I'll show you a little later about the quantitative comparison in the PCR. So, and yeah, I mean, so the universal, the specific signals are very specific, like either it's there or it's not really. The universal signal is a little bit contaminated, and because every preparation of TAC has some amount of bacterial DNA from which the enzymes were grown in, and so we know about those kinds of things. In terms of the sterility, so the beautiful thing about ventilated patients, and the sad thing for them, is that they're intubated. That's really great for doing this, because that means that, so the endotracheal tube goes down about to here. So what you're getting is, and that's sealed, right? There's an inflatable little cuff. So everything that comes up, that comes into that HME, has to come up through that endotracheal tube. So it's not coming from the mouth. So stuff that's like contaminating oral fauna that would be sort of confounding potentially, not really an issue. And you know, with your mucociliary elevators and all that kind of stuff, typically your lungs should be sterile, right? So the purpose of this, later I'm going to blow your mind, okay? And then there won't be like, I think the kinds of process things that I'll show you will be much more convincing than these static head-to-head comparisons. But the first thing that you have to do in any setting is to show that, like, you know, first of all, are we detecting organisms there? And do they agree with the kinds of things that we find in another quantitative assay, right? So this was kind of the first thing to sort of say, hey, okay, are we on the same page or not? So here's some statistics associated with those data. The sort of central panel that goes across the bottom shows you the breakdown by organisms of the contingency tables comparing BAL versus HME detection. And you can see that, for, you know, we missed some fecalis and some aureus, but for the most part, so these are 99% confidence intervals that I've shown you. For the most part, detecting by organism is pretty much uniform. And we spent a lot of time, actually, I'm not going to tell you about all these things, but I spent a lot of time working out protocols so that we would uniformly disrupt all the organisms in these samples, both gram-positive and gram-negatives, that have different propensity for being broken apart and having their DNA extracted. We spent a lot of time developing protocols that were maximally unbiased. And that's kind of a topic du jour. If you read sort of applied and environmental microbiology, that's an interesting sort of field and stream project and comes into play in these kinds of things. So the bottom line is, if you combine the targets and look at sensitivity and specificity, it's really pretty good. And here's the quantitative correlation between, on the bottom, the detection from the HMEs with the detection by the BAL. And I think all in all, we were pretty happy with that. I'm convinced. So then we went on to look at what else is going on in these filters. And we were also thinking about this is motivated by the desire to normalize. How do we normalize when these people are breathing up? Everybody exhales things differently. How, you know, normalizing BAL samples has been an issue for a long time. There's variable dilution. I don't want to spend a lot of time because I got lots of other fun things to tell you about. So we thought about using surfactant protein B to normalize things. Surfactant protein B doesn't change its amount over lots of comorbid conditions. This has been shown in a study of children. So I thought SPB was good. The most important thing is we found SPB. We also found DPPC, which are the two, that's the biggest protein and lipid component of surfactant. So there's, what I believe is happening is small surfactant droplets are forming in the lungs. These surfactant droplets are being aerosolized. The distribution of these sizes is large enough to encapsulate bacteria. Every once in a while they do that, and these things come up. So basically anything in your lungs that's miscible in the, in the surfactant, in that alveolar lining fluid, those things are going to show up in the filter. My focus on dynamics, I'm always interested in time series, and infection is a process. We all know this. Causality involves precedent in time. I don't think I need to tell anybody about causality here. I think you all know about causality. I don't know anything about causality, but it seems to me dynamics is king. So here's a picture that I show because of, this is the state of the art in pneumonia work. And I think one of the reasons why things might be slow. So this is a recent paper, a high-profile paper in PLOS One. You know, they reject my papers, but they publish this stuff. And the top graph, you'll see, the top graph, what they did is they had a cohort of 16 mice, 16 different mice, right? So this is different mice. They put some of this pneumococcus up their nose, right? They infected the 16 mice, and then they sacrificed 4 mice per time point. They took their left lungs, ground them up for their kinase experiments. They took the right lung, that part is clever, I think. They took the left lung for one part, they took the right lung to assay for the number of bacteria, and here is the growth curve of the bacteria in that colony of mice, okay? And then they did all kinds of looking at which cytokines are expressed in response and so forth, and that's really pretty cool, right? But this is a gestalt, right? This is a gestalt of a colony of mice. It's not looking at one mouse. It's not looking at the infection process in one mouse over time, right? And then getting 16 reproducible bits of statistics of time courses, right? It's some kind of weird mixed thing that, I don't know, confuses me. On the other hand, this is repeatedly profiling the HME filters on a single patient over time. Because these things are thrown in the trash every 8 hours, I can look every 8 hours in a patient and I can look for whatever the heck I want to because no one's going to stop me. And that's the best part about being me, because no one's, so right now, tomorrow I'll be living on the street and then it's going to suck to be me. But for right now, it's all good. So this shows you the antibiotics that were administered by the physicians, and it shows you the black dots show the accumulation, the log number of copies of Klebsiella pneumonia that we found. The red dot is when a BAL was performed, and the amount from the BAL. And the dotted line, we looked at enterotoxin, okay? There's a nice little clam assay that you can do to assay for enterotoxin, and so we applied this assay. And I don't know what you think, but my take-home message here is when they put the tube in the person to do the BAL, they stirred up the hornet's nest, right? Because if you look, the enterotoxin goes bananas after the production of the BAL. And that makes good sense because they're taking 100 cc's of fluid in each lobe of the lung and they squirt it in there, right? It's nice, well mixing. And this is far more convincing because these are time series and what you see is the bacteria going up. That's no artifact. Well,
did they apply VANC? Is there a time point, or what does the bars mean for the VANC and the?
That means that over that time course, they were administering vancomycin.
Okay, so they gave it at time 0.
That I'm not. That I cannot certify that. I don't know if you know when in the past they started. I don't know, but I do know that vancomycin was continually given over that course.
So this means that like basically the bacteria basically ignored it.
They ignored it, yeah. They ignored it. So what I've learned, again, I'm not a physician, but I've learned some cool stuff. And one of the things that I've learned is vancomycin is not a very good drug in the lungs because it doesn't penetrate the lungs very well. For some reason that I don't understand what the biophysics of that is, but it's true. Some antibiotics get into the lungs better than others. And vancomycin is a very poor one. So here's another one. This actually was the first time series that we had. And what happened here is you can see the black dots are again us profiling. Here, this is a universal. We're plotting total bacterial load. At the green dot, this patient was extubated. And at the red dot, so this patient subsequently crashed. They reintubated the patient. So they didn't have any of this data when they did this, right? So at the green dot, they extubated the patient. They immediately reintubated the patient because that person crashed. The red dot, they performed a BAL, and then they started antibiotics at that time point, okay? And now what I'm going to tell you about is that in this case, I can calculate what's called an early treatment gain, okay? So for instance, had the physicians started these antibiotics 1 day earlier at the green dot instead of at the red dot, this patient would have resolved 2.4 days earlier. That's the treatment gain, okay? So here's what I'm telling you. So you're not as convinced when I show you the comparison head-to-head of these organisms in BAL versus the organisms in the filter, right? But what you cannot argue with is the thing that makes me believe that these are real are the fact that these bacteria go up without antibiotics, and when you give antibiotics, they go down. That's real, right? That's not an artifact. That's I don't know what you call that, you know, like, but as human beings, our brains are always looking for evidence that tells us whether we're on, you know, what's that called? Like, you're the machine learning guys. You tell me, what's that called when you look for additional supporting evidence that the universe, like, that things are making sense? You know, I don't know, like in math, it would be like dimensional analysis. You know, like if you get cosine x equals 4, you know you screwed up, right? Because cosine's between -1 and 1, right? But here, like, this is like, I don't know, it's correlation, but it's tells you that somehow.
So this is detected in the filter, right?
Yeah, the black dots are all in the filter.
And which bacteria it is in that graph?
So here, what I'm showing you is the total amount of bacteria, not differentiated by species. It's just the total bacteria. So in terms of early treatment gain, so I'm going to spend a little interlude here. This is fun for you now. This is going to be a little fun math interlude. So how can we calculate an early treatment gain? How can we look at these time series and estimate what would have happened had I given antibiotics sooner? Okay? Now, the beautiful thing here is that, so in general, what they'll teach you in medical school, or maybe in pharmacy school or something like that, is like, if you give a certain amount of antibiotic by weight, you know, and this is worked out how? In a bunch of mice, right? Not people, right? So what's the pharmacokinetics of vancomycin? You know, it has nothing to do with you or your genetics or anything like that, right? It's got to do with some cohort of mice, maybe if you're lucky, and most likely they grew a bunch of organisms in a petri dish and did pharmacokinetics in vitro, right? So here, what we're talking about is, like, if I look at that regression line here and I calculate the rate at which those bacteria are washing out in response to those antibiotics, that's in the patient background, right? That's as close to personalized medicine as you're going to get. So here, the idea is very simply, so we're looking at the rise in the bacteria. In the natural course of events, antibiotics were administered at some time, and the bacteria declined with some rate m. Okay, what would have happened had I applied antibiotics earlier? Right, then there's a relationship between the earlier treatment time E to the amount of resolution time T, right? And that ratio is actually given by this gain. And the gain from this picture, you can actually do a little similar triangles, and you can figure out that the gain is given by the ratio of those rates plus 1. Okay, so the gain is always bigger than 1 in this particular setting. And we wanted to think about this in a more general mathematical way, not just in that sort of simplified picture. Although the simplified picture is kind of, I mean, that represents the sort of the complexity of the data. But we look to model treatment as a pair of dynamical systems. So X here represents the bacterial load, Y represents the immune response. That could be the number of PMNs, or it could be however you want to categorize the immune response. So this is the growth of the pathogen in the person prior to antibiotic treatment. That's this pair of equations. Okay, and then the second model, which is subscripted with an A, that's after antibiotic administration. Okay, so that's how the dynamics are progressing after the administration of antibiotics. Okay, so that vector field is subscripted by A. And so here's going to be the notation. T0 is the time at which you got inoculated. X0 is the inoculum size. At time T1 is when, which is bigger than 0, bigger than T0, is when the physicians treated the infection with antibiotics. Chi is the load, the bacterial load that you would consider to be resolution. T is the time that it takes to get to resolution. And then the gain is defined as the rate of change of that resolution time with respect to the treatment time. Okay? So it's possible to calculate a general formula, and this is a really quick derivation. So the load after treatment, by using the variation of parameters formula in the antibiotic administered system, we can get a formula for the resolution time and the value at resolution. And if you differentiate this with respect to the treatment time, use Leibniz's rule for how you differentiate under an integral, we can get a relationship here. And you can solve this for the gain to get a nice formula, sort of general formula. For specific examples of differential equations, this is either more or less complicated. What kind of things can we learn from really simple systems? So let's say we consider really simple linear models. So here we have a model where the bacteria are growing at some constant growth rate, and then after antibiotic administration, they're declining with exponentially with another growth rate. In this case, we can figure out, we know what the solutions are. It's very simple. We set a resolution equal to chi, and we solve. And the treatment time, you see it's linear. So the resolution time is linear in the treatment time. And the gain is given by exactly the formula that I showed you that you can deduce geometrically from the picture for exactly the same setting. Okay? So this is the exact same formula that now we worked out from differential equations algebraically, versus you can work out the exact same formula geometrically. And it tells you that basically the gain is 1 plus the ratio of these rates of the slope going up versus the slope coming down. So we can also get a formula for this in higher dimensional linear models, and it collapses to the same formula. The logistic model is informative for the following reason. In a logistic model, you've got a carrying capacity that limits. So in your body, bacteria are going to have a hard time finding iron. They're going to have a hard time finding carbon sources or nutrients. And so their growth is capped at top, right? Just like in nature, if they're growing on a grape, eventually they're going to run out of stuff, and their growth is capped. So what can we learn about the treatment gain from looking at a logistic model? If you look at a logistic model, what you get is you see that you have the gain is the same as in the exponential model, except you have this extra piece. And what does this extra piece tell us? Which is the ratio of the amount of bacteria at the treatment time over the carrying capacity. What's interesting here is that this tells us that as the bacterial load, if you're treating close to the carrying capacity, your early treatment gain is going to collapse to 1. So it's going to erode. So your power, the benefits of early treatment diminish the longer the infection remains untreated. Okay? And that's from a very simple model, but it's a fairly powerful indication of the truth. And then we can look at a slightly more complex situation where we add an immune response, but in a simple way. Imagine if the immune response is a program response. Imagine that, that growth prior to surveillance is exponential or logistic even, and that an adaptive response is triggered at some fixed pathogen level. So the human body is able to detect, once there's 10 to the 2 or 3 bacteria, then your body goes into action, right? Then your adaptive immune system goes freaking bananas. Says, this is not supposed to be here. Let's recruit some PMNs into this lung. Let's kill these guys, right? And as long as once that program is triggered, that program happens in its own time, happens at its own pace, basically independent of what the organisms then are doing in terms of their density. It's a program triggered like a reflex loop, right? It's a reflex loop. And to the extent that the real immune response is like a reflex loop, to that extent, this model will capture the behavior. And in this model, again, you can calculate that the gain. And what's interesting here is that now the gain depends on the immune system, on the value of the immune system at the resolution time. Okay? And this is interesting for the following reason, because the size of the treatment gain is influenced by the asymptotic efficiency of the immune response, right? So if the immune response is larger than the growth rate, so that if the immune response itself would have eventually taken care of the infection by itself, okay, then the treatment gain is less than 1, okay? It erodes. If not, then the treatment gain is bigger than 1, okay? So, and also, if we assume that the immune response is kind of a monotone function, like a sigmoid, right? It's some approximation of a step function. So it's like off and then it's on. And if that saturating value would have killed the infection, then your early treatment gain. But again, as Y increases, your efficacy in treatment, the later you wait to treat, the more it erodes your treatment gain. Okay? So we're working on these. We think that this is an interesting and powerful approach. I love calculating these early treatment gains. We're looking at more realistic predator-prey models. And also, the real goal here is to be able to look at these time series, like I'm going to show you a few more time series, don't worry, and to be able to take those time series and actually regress early treatment gains as rigorously as we possibly can. Because what I would like to argue is this, right, is that you now have a way to look, you have a way to surveil the patient well before you do a BAL, right? So you have a way to try to predict whether someone's going to get pneumonia or not, and you can actually treat someone earlier, much earlier than in the past. And so predicting what the treatment gains would be has value and supports my position as a politician looking for money. Yes?
So in terms of modeling the immune system, so you're essentially just modeling whether, so do you know what, if a patient starts to have an upward profile of bacteria, do you know what it looks like when the immune system starts to try to take care of that on its own?
So I can look at the same data that you could look at. So for instance, there's, so the rest of the talk is devoted to what I can mine out of the things like biomarkers and things like that. But the other things that you can get is, for instance, you can get white blood cells, you can get temperature, you can get, whatever it is that you can measure, right? Problem is, the real problem and the beauty of this is that pulmonary stuff is highly compartmentalized, right? You don't want to get sepsis, you want to keep everything localized. And that people have had very poor success with finding biomarkers for pulmonary infection from the blood because of compartmentalization. The deal here is this reservoir is so important because it's from the actual compartment. But you can look at like things, so heart rate variability, right? There's all kinds of measures of things that you can look at.
I guess I was asking in terms of like how the immune system attacks the virus or the bacteria. Is it modeled in the same way as how when you give them antibiotics, it's literally like a linear, log drop, or is it a little bit more? It's less sort of reverse exponential in how it kills the bacteria.
Yeah. So the predator-prey interaction between the bacteria and the immune system, I mean, that's a predator-prey dynamics. It's clearly, I think it's highly nonlinear and very complicated. And that is one thing that Todd and I are actively, we have a paper about early adaptive immunity and models for that and trying to understand that, and then also trying to base that upon data that we can measure. So here's an example of a patient who's loaded with Staph aureus. It turns out that this was a case of methicillin-resistant Staph aureus, and it's also the last line of defense against Staph aureus is not necessarily vancomycin, but it's this linezolid. And this patient was also linezolid resistant. And what's so interesting is the initial growth rate of these bacteria in the person, the doubling time is about 3 hours, which is extremely fast in a person. It's slow if you're talking about 20 minutes in a flask, but in a person, that's very fast. There was a very rapid rise in this person. They were able to sort of control it, and then the bacteria rebounded. The patient eventually died. What I'll draw your attention to that I find so interesting in this data set is this, is that when in the early part of this infection, what you'll see is the data points don't hug those lines, right? And I'll show you data later on about a hypothesis about why that's true. But what you see is that later in this curve on the rebound, those data hug that line really well. Now, if I were to take a bacterial growth curve in a culture in my lab, like if I grew yeast or E. coli or something in a flask with just in glucose broth, okay? Those bacteria would grow exponentially or logistically, and those data on regression would hug that line. And that is because they're growing in rich media and they're uncontested, okay? So what's going on here, you see that there's a marked loss in complexity of that signal, right? Just like loss of complexity in heart rate is an interesting thing. What explains this loss of complexity? And I think it goes like this, is that in this patient's immune system has collapsed. Also, there's been the purifying selection of these drugs over time, which means that you don't have a culture of cells that are very heterogeneous, right? These are highly, highly homogeneous drug-resistant organisms growing in a rich environment, and they're uncontested. And that's why you see that loss of complexity. So the final sets of data that I'll show you, this is another patient that was dosed to the gills and was infected with Staph aureus. And the next curve shows you, so here, these black dots represent Staph aureus that we've mined from the filters. Those are the exact same data points that have gone to construct this dotted gray line. Okay, so the gray line is Staph aureus. This patient, in addition to that, also was subsequently infected with Pseudomonas. Okay, and the Pseudomonas is shown in the dotted black line. Okay, so the Pseudomonas infection came out. And I don't know if you can see there, but just after the rise in the Pseudomonas, there's a little blue line. That's the time of the BAL. See back here, the red dot is the BAL at about 300 hours or so. That's this little blue, that's marked by that blue line. And each of these 6 panels is where we've profiled for different cytokines from the HME, okay? So these are different cytokine spikes, IL-1B, GCSF, which recruits PMNs, and all the different IL-6, et cetera. And what you'll see, I used to be kind of mad at my postdoc when he showed me these PCR data, because you see those dots are all over the place. They do not fit my beautiful regressed lines, okay? And that, if you're, like a freak show like me, that makes you mad. And so my technician heard about it, or my postdoc heard about it. And the deal is, though, when I saw this curve, then I relaxed. And I'll tell you why. Because look at every place that the bacterial growth rate changes. There's a cytokine spike that's right there, right? So initially, the bacteria right here in the beginning, I don't have a laser pointer, but in the beginning, the bacteria are growing rapidly. There's a cytokine spike, and they slow down, right? Then the bacteria start ramping up the cytokine spike, okay? So there's a game. There's a game that's going on here. This is not coincidental. And if you take a linear combination of the bacterial signals, that's the black line. And you can see that that black line sort of forms the letter M, right? And if you take a linear combination of the cytokine signals, you get the red line, and that kind of forms the letter W, right? And if you formally sort of, and the green dotted line is where the BAL was performed, right? So if you look at the correlation between these, you see that they're anti-correlated, right? And that's what you expect in a predator-prey game. They're in opposition to each other, right? They're playing a game. The reason why, I don't know if you've seen this. Have you seen this TV show? It's called Restaurant Stakeout. This guy goes into restaurants and he puts up video cameras and he sees all the waiters doing bad behavior, and then he can tell the owners of the restaurant like who to fire and how to change the business so that they don't lose their. It's called Restaurant Stakeout. It's great. There's a guy here in town who's got these Uncle Jack steakhouses. That's the guy. Willie Deagle's the owner. And in the show, like this is on Food Network, and in the show, the deal is it's like they show Willie, who's kind of a big guy behind a desk. He's Italian. And they're like, restaurant owners around the world are reaching out to Willie. And, Willie's coming to save their business. And it's like he's a mafia guy, right? They're reaching out to Willie, and he's going to, and in very much the same way, when I saw these data, it came to me that I needed to reach out to Constantine. Because the deal is you look at this and you say to yourself, look at these signals, right? There are signatures in here, right? Alex, his whole thesis was talking about signatures, right? There are signatures of infection in here, right? There are sequences of events. I guarantee you in a patient that's infected, that's going to develop a pneumonia, that's going to have, like for instance, IL-6, that spike in IL-6 precedes the BAL by about 72 hours, okay? IL-6 is known to be a potent inducer of fever, right? It's got some pyrogenic deal with your brain. Google it. I did. That's how I found out, right? I just Googled IL-6, and everything you want to know about IL-6 is right in Wiki, right? And it's a pyrogen. IL-6 is a pyrogen. So, I'm speculating here, right? But that's my job. That's my brain. It says, look, there's that IL-6 spike. They probably saw a fever, a little bit later. So there's signatures. There's a process that's happening here. And the idea is that, Constantine has has developed, and Alex together, and all y'all have developed these tools that allow you to mine the relevant features, right? So what we need to do from these kinds of time series is to mine out what are the signatures that precede infection and differentiate those from people that don't get infected, right? And that, in that way, try to inform treatment, right? Early treatment. Develop, you know, the idea would be to develop, what needs to happen now is to develop a protocol that you can go and build a trial around, right? That you can either implement it or not implement it. And then you can see, does that shave a ventilator day? Does that shave, you know, something off of a person's stay?
And the signatures will be measured from the repeated in-time measurements of this biomarker panel, right?
Exactly. So I'll show you a few more. So we can get good genomic data out of here from the HME. You can extract the bacteria. You can get RNA. You can look at genes. The other thing that we've done is we've done proteomics. So on the fluid. So the way that we capture, we have the HMEs. We run the fluid through a filter to capture the bacteria. So we can take the bacteria and look at what's going on with them. And in the flow through are all these other things like human proteins that are part of the innate immune response, right, that are secreted by the alveolar epithelial cells and the macrophages and whatever, and all this stuff is miscible in the alveolar lining fluid, and that stuff comes up. And what we've demonstrated now, and we've looked at a time series of these, and this was a preliminary thing that we looked at, that you can find all kinds of classes of interesting molecules, potential biomarkers of infection that show up, like neutrophil defensins, lactoferrins, things that, all of these things. So, and I'm not going to bore you anymore with any of this. Let's get it over with. So I really need to thank Constantine and Alex for allowing me to come here and give me this opportunity to talk to you today, tell you about my stuff, and also, you know, try to reach out and get their help. I really appreciate that. I need to thank my clinical collaborator, Addison May. Addison runs the surgical and trauma units at Vanderbilt, and without that guy, I wouldn't have samples. Dan, my former chairman, who, you know, gave me $2,000 of his own money to get a microfuge. Gotta love that. Rick, who put up with me for about 8 or 9 years, like freeloading in his lab, and Gordon and Victor, who gave us support and money. I need to thank my collaborator, Todd Young, and I really appreciate you as an audience. Thank you. Yin.
So do you think, so I noticed you mentioned this idea of, you know, when they do the BAL, it sort of stirs up the hornet's nest, the endotoxins start going crazy. Do you think you can make a case that, like, before they do BALs, they should load the patients just with a spike of antibiotics or something like that, and sort of, you know, try to preempt that spike? Or well, first of all, is that spike bad? You know, I mean, is it causing the patient to, you know, have a fever that spikes or puts them in a more unstable state where they're at risk of, you know, having sort of cardiac events or something like that? And secondly, you know, do you think there's value in actually preventing that spike from happening?
So the protocol which they have in place at Vandy is this, is that when they have suspicion of pneumonia, they perform a BAL and they give them empiric antibiotics. They have an empiric antibiotic rotation, so at all times they have 3 antibiotics that cover that, you know, it's broad spectrum coverage, so they don't know what's there because the micro results haven't come back. Those take about 72 hours, right, or 48 to 72 hours. So they give them broad spectrum coverage because of the suspicion. And then they are supposed to, if the results come back, either tailor that specifically to the organisms with known, you know, from the BALs, when they culture the organisms, they do the sensitivities. So they can have ideas about how to specifically tailor antibiotics. And also they should wean them off of the empiric coverage if they don't find organisms. So that's their strategy. Yeah, I mean, squirting fluid in there is going to stir those things up. It's going to shed exotoxins. Is that going to cause an inflammatory response? Probably. How severe that is, I can't really speak to. I was just hypothesizing based on what we saw there about what's going on. And I'm not advocating any treatment protocol in that sense, but that's The BALs are invasive. That's the beauty of the HME, and what I would argue is that, what we're looking at is how complete a reservoir of information is it? It's got every kind of information that we've thought to look at. It seems reproducible. It's totally non-invasive. Will it replace BAL? Probably not, but it can definitely augment BAL in ways, you can't do, no one does repeat BALs. I mean, you do repeat BALs when you think down the road something's really wrong, but you don't do them every day.
So why you can't replace BAL?
Because it's hard to let go, right? I mean, if the HME turns out to be a reliable, reproducible source of information, then you could potentially let BALs go. But human beings, hold on tight, right? I mean,
in principle, you can even have some kind of dynamic new filter that is optimized for this, because filter is for different reasons. It has some sensors.
That's what we're thinking, is that you can develop a point-of-care device that can do these kinds of, that could do the bacterial or cytokine or even other biomarker detectors. And it's, so we have some ideas about how you would design something like that. I think it's conducive to that. I think you could do that, and I think you could do it for, I think right now they can make those HME filters for nickels and sell them for $10. I think you can make a sensor that's going to cost you $8 and sell it for $10. I mean, it's not the same profit margin, but yeah. See, that's why you have a better head for business than me.
Well, I think the physicians do get paid, right, when they do a BAL?
Oh, yeah. So that's the thing. So it costs $700 in the outpatient setting. I have a friend who's a coding auditor told me that. A BAL is billed at $700 in the outpatient setting.
But they aren't going to get reimbursed for that necessarily, because ventilator-associated pneumonia doesn't get reimbursed under Medicare anymore.
Oh, got it. Is it a never?
That's your own fault. So you cover it.
Right.
So that's the hospitals who pay, right?
Yeah.
Yeah, they don't, the insurance won't. So the early treatment gain, that's one of the things. This is a grant writing thing, I mean, that's a model that's strongly supported by the payers, that you would like to treat early and make sure that things. The other thing is that one of the things that I didn't say here, which I think is really true, is that there's a lot of discussion about ARDS, and you can clear up someone's infection. The older you are, the harder it is, right? Age is, unfortunately, like me, I'm getting older, and you're starting to realize that I'm not 16 anymore, right? I mean, it sucks. And that's really, if you look at this, age is a risk factor for these kinds of things. Our immune system is not as responsive. And the deal is if you get a pneumonia, that costs you something. And lung damage is, while, we figure that, your cuts heal, but they heal slower, right? And these kinds of things, having a bacterial infection, having a protracted immune battle against them, it can lead to chronic stuff like ALI, ARDS. So there is value in trying to treat bacterial infections as quickly and as efficiently as possible to try to minimize those kind of collateral damage. I think that's a reason that you can make I'd like to make an argument for that.
Do you think this would work outside of the a ventilator? I mean, so if patients come into the hospital and it seems like they have something going on with them, and you can just have
this like nose,
like a breathalyzer. You can just breathalyze into the sucker and it tells you something.
So it's clearly these aerosolized droplets. The problem with just breathing into a tube or something like that to detect walking pneumonias or all kinds of things, right? The problem there is you're going to get this influence of oral stuff, nasal carriage. But one of the things there's a guy at Vandy, Art Wheeler, who's, I don't know him very well, but he runs the medical intensive care unit, and he's a very clever guy, and he said the following thing to me, that, for instance, if you detect Mycobacterium when someone's breathing in a tube, that isn't supposed to be in your face. So regardless, you know what I mean? If you find that finding tells you something, so there's something like that. And there's maybe other things that we haven't really taken that far. I've shown you all I got.
It's awesome.
Thank you.